at DTU. I would like to welcome everybody at this doctoral thesis defense at DTU. Especially, I'd like to welcome our doctoral candidate and give him a big round of applause, Professor Andreas Hougaard, Laustsen, Kiel. And our two official opponents, Professor Brian Fry, with us today, thank you very much, University of Queensland, Australia. And Professor Michael Hust, TU Braunschweig, Germany. Of course, a very warm welcome to our own Professor Peter West, Institute of Bioengineering, DTU. He is our chairman of the assessment committee. Now, the proceedings are as follows, and I should maybe say my name is Camilla Hougaard. I am the Dean of Sustainability of this university, and I'll be your chairperson of today's proceedings. Now, also I should say that this is recorded for purposes of documentation. The proceedings are as follows. After this brief introduction from me, the doctoral candidate, Andreas Hougaard, Laustsen, Kiel, will present his thesis, Engineering Antivenom, and the lecture will be approximately 30 minutes. This is followed by a short break in which you may register with me or Dide. Dide, you can raise your hand, please, if you wish to act as an unofficial opponent, either ex-auditorium or from online, during the proceedings. After the break, I shall invite the official opponents in turn to discuss the thesis with the candidate. Mindful of the time difference, of course, Professor Brian Fry goes first. Now, should any of you become unofficial opponents, and if you have registered correctly, subsequently you will be given the chance to examine Andreas individually for a maximum of 20 minutes each. After these discussions, the formal part of the defense is over, and DTU will host a small reception outside the lobby, and you can follow me. At this point, I would like to invite Professor Andreas Hougaard, Laustsen, Kiel, to the podium, please.
Thank you very much, Camilla. Thanks to the committee for assessing my dissertation and of course showing up either online or in person. And then thanks to all of you for showing up as audience. I've been looking very much forward to this day, and I will of course talk a bit about all the work that I've been doing in the last 13 years, starting out by giving, you could say, an overview of what is the intention, what was the sort of vision of it, and then go into a bit more technical details of the main, you could say, conclusions or findings of the work. But it all starts with snake venom. And important to know is that snake venoms are not just, you could say, the same. They don't consist of just one toxin. They're actually quite complex mixtures of protein-based toxins. And given that they are these complex mixtures, they need to be neutralized with complex mixtures of antitoxins, in this case depicted as antibodies. And you could say the vision of my work has really been to say, can we do something new, something more modern with antivenom? And can we go in and design mixtures of broadly neutralizing antibodies, express them recombinantly, and use these as, you could say, a new generation of antivenoms? And I'll get a bit back to why that is. But before jumping in to, you could say, the science, I want to take you back to Tanzania in 2011. I was on safari with my family. We were just there to see animals. But we came across a snakebite clinic where 2 children had been bitten by spitting cobras. And these children were amputated either by around the elbow or the knee. And I had expected that even in Africa, you would have received better treatment than amputation if you were bitten by a venomous snake. So this, of course, left a strong impression with me that I thought, why is it that antivenoms are not available, and why are people still affected by something that has been around for as long as mankind, snakebite? So there's going to be some quite nasty pictures, just so you're warned. But as an example, here's another child, not the same child as those that we saw, but another child who's also been bitten by a spitting cobra. And what has happened here is that you can see the venom has caused a lot of local tissue damage to a point where it will affect mobility. And the reason for this is that spitting cobra venoms, they have a lot of so-called cytotoxins that go in and interfere with cell membranes and cause cells to disrupt. And this leads to these quite nasty disfigurements. And that's, you could say, one example of what some snake venoms can do, that they can cause a lot of local tissue damage. Another example, also a child who's been bitten by a snake with hemotoxic venom. In this case, some toxins have gone in and chopped up the capillaries, meaning the blood vessels, and then a different set of toxins have gone in and interfered with the blood's ability to coagulate, causing this hemorrhage as we see here. And that's also a typical clinical manifestation of some snake bites. And the last example is a child who's been bitten by a neurotoxic snake, where neurotoxins have gone in, bound to the nicotinic acetylcholine receptor responsible for neuromuscular transmission, and simply blocked the child's ability to move. So it's paralyzed, and if this child was not helped and received antivenom and was ventilated, the result would be that it would stop breathing simply because the muscles around the lungs would stop working. So clearly, snakebite is a serious medical emergency for the individual. That's probably not a big surprise to many of you, at least if it's untreated. But if we zoom out and look at it at a global scale, it's a massive global health issue. So snakebite causes around 2.5 million envenomings per year. Somewhere around 100,000 people die, and about 3 to 4 times that number are left with permanent damage, such as amputated limbs. Snakebite is categorized as one of the 21 category A neglected tropical diseases by the WHO, which means that they are the worst neglected tropical diseases, but snakebite alone kills more people than the 20 others combined. If you take UN's Sustainable Development Goals, where they have different ambitions of reducing neglected tropical diseases the deaths from neglected tropical diseases with 50%, that's unachievable if nothing is done to snakebite. So it is really a massive, massive health issue, just not so much in the part of the world where we are right now. So if we look at venoms, there's perhaps one important thing that all of you should take away from today, is that there's a difference between venoms and poisons. We don't differentiate in Danish or German, many other languages. But venoms, they're based on proteins, which means that they cannot be absorbed in the stomach and they need to be injected. This is the reason why venomous animals, they have stingers or fangs, whereas poisonous animals, they have typically small molecules somewhere in them that can be absorbed in the stomach, which means that you will be intoxicated if you eat a poisonous animal. But you could say that this is important, that eating a venomous animal is probably not a problem. But it also has another important implication. And that is, because venoms are protein-based, it's possible to use them for immunization. And this is how antivenoms have been made for 130 years. You simply take the venoms from the snakes. You milk the snake, you take the venoms that you want to make an antivenom against. You then inject them into a large animal, typically a horse, and you do so for a year or a year and a half. And after this period of time, the horse's immune system will have made a lot of antibodies that can then be harvested via large blood samples, you could say, where you send the red blood cells back to the animal typically, and then use the serum or plasma to isolate the mixture of antibodies and formulate them into an antivenom. And isn't this just perfect? Well, I think it's important to say that antivenoms are still instrumental in saving lives, and they were a major breakthrough when they were invented 130 years ago. Really, it's impressive to see a medicine, you could say, on the market that has been around for more than 100 years. There's simply been nothing else to replace it yet. That being said, when you take something from an animal and inject it into a human, there's no question that there's issues around that. The human immune system might recognize these horse-based these horse-derived antibodies as foreign and react against them. There's a limitation for how efficacious you can make antivenoms. They don't necessarily always have broad enough neutralization or have high enough affinity against their target, so you need very large doses. And this manufacturing technology doesn't scale so well. If you need the double amount of antivenom, you need the double amount of horses. So there are some economies of scale that could be better. And this is also, you could say, why I believe that in the future, antivenom should be made in the same way as most other therapeutic proteins are made, using cell culture or fermentation, where you take the genes for the antibodies you want to make, put them into cells, and grow them in a bioreactor, and then formulate them into an antivenom after some purification steps. Just like blood factors have been made for decades, and insulins, and many other proteins. The benefit with this would be that you could make human versions of the antibodies, or at least versions that are compatible with the human immune system. You could improve their pharmacokinetics, you could improve their neutralization capacity, you can avoid being dependent on snakes and horses and have that batch-to-batch variation. And I also believe that you could have a more consistent and low-cost manufacture, especially in the future. And I also think that if you could make safer products, it wouldn't just be a benefit for safety itself. It will also allow clinicians to use antivenoms much faster in the field. And snakebite is a medical emergency where the quicker you get treatment, the better efficacy the treatment will have. So I think indirectly improving safety, you'll also improve efficacy. And that's sort of, you could say, the whole background for why do I think it makes sense to go in and replace these plasma-based products with a new modern biotechnology-based solution. And to do that, you need to look at the venoms. So I'm not going to go through a big comprehensive overview of snake venoms, but I'm going to take you through an example. The black mamba, a snake venom or venom from a snake that I worked a bit with. This is a very notorious snake that many of you probably heard about. It's Africa's largest venomous snake. It can reach 4.5 meters and look down at a short Viking like myself, because it can raise its body half off the ground. And it has a very fast and potent acting venom. It's known for always biting multiple times, always injecting venom. It can slither faster than our fastest 100-meter runners can run. Brian might disagree. At least many herpetologists say that there's no venomous snake that's aggressive towards humans. Those that live close to this snake's habitat will probably say that then it's at least proactively defensive. It's a bad guy. And in short, just to give you an example of how you look at venoms and to sort of say what is the basis for looking at venom as a drug target, what I did in my PhD, so this is before the stuff that's being evaluated today, I took the venom and separated with HPLC into its different components, used proteomics to identify what are the toxins, how much of each toxin is there, and then introduced a rather simple concept of a toxicity score, looking at if there's a lot of a toxin and it's very potent, it gets a high score. If there's not so much and it's not so potent, it gets a low score. And then I allow that score to sort of guide whether a toxin needs to be neutralized in an antivenom. Is it simply medically important enough? And that led to this, my first scientific paper with a very short title. That is, you could say, kind of the first example of the framework that we've been using in my group later on to analyze venoms. And to sort of fast forward a little bit, more recently, my group has worked with all the most medically relevant snakes in Africa, and here I've just highlighted the 18 elapids, so that's the cobras, mambas, and ringhals. And we essentially used some updated methods where Carol and Yang, my former postdocs, led a study where we analyzed these venoms and could sort of map what are the compositions of these venoms and what toxins do we need to go after. And if any of you are interested, here's the paper, and you can see it's already one line shorter. So that's, you could say, we can discuss more about venoms in the Q&A, but that's, you could say, the basic framework for how do we look at venoms when we view them as drug targets. Then moving into antibody discovery, I want to touch upon the main technique that me and my group have been using over the last few years. It's called phage display. And here we make use of, you could say, the link between phenotype and genotype by having antibody fragments displayed on the surface of a phage particle. And in this phage particle, we have its DNA plus the gene encoding for that antibody fragment. So that you could essentially view this antibody fragment as the product, and here's the big barcode you can sort of scan with DNA sequencing and figure out what is the amino acid sequence. And the smart thing about doing this is that you can make very large libraries of different antibodies. And the company Iontas, led by John McCafferty, were kind to donate their big human monoclonal antibody library to my lab that we've then been working with. And having such a large library means that when you introduce a toxin or any other type of antigen to this library, there's a large chance that you'll have something in there that will bind. So the way you use it is you take your, in this case, toxin of interest, you stick it to the bottom of a well or a bead, you pan the library on top. As said, statistically there will often, almost always, be something that binds. What doesn't bind, you wash away. What does bind, you elute, you amplify it in cells, and then you often let what binds compete against each other, so you don't just get binders, but you get the best binders. You let the binders compete. And when you're satisfied, you continue and analyze the output with methods such as ELISA and DNA sequencing to figure out what antibodies actually bind these toxins. And I'm going to go back to the black mamba because that was also the snake venom that I used in perhaps one of the most important studies I did for antibody discovery. Here we had already identified the toxins that we needed to neutralize, and we brought it through this phage display selection process, had outputs that were enriched for binders, screened different monoclonal antibodies, sequenced these and found some unique ones, and then we converted them from the scFv format that you use in the library to the full regular human IgG format. And as you see, most of them retained binding. And the more interesting thing is, of course, that we then took some of the best antibodies and mixed them in 2 different cocktails, and could show when we mixed the venom and the cocktail and injected it intracerebroventricularly in mice, that we could save mice from dying in a dose-dependent manner. And I think this was perhaps the first major breakthrough, at least in my work, in the more like antivenom research part. But in this work, we had also worked with other toxins from this guy, the monocle cobra from Southeast Asia. And we had found antibodies against the main toxins from this snake. You see how they perform here. You see that when you mix them with toxins and inject it into a mouse, mice, they do live longer, but they still die after some hours. So you could say they're not good enough. So my former PhD student, Line, she used a method called light chain shuffling, where you, in short, take your heavy chains of the antibodies and exchange it with a library of light chains and find a better light chain partner with the ambition of either finding different properties or better binding, higher affinity antibodies. And that is exactly what she then succeeded with, and showed that here we see that we can actually save most of the mice when we have exchanged that light chain in the best antibody. And here's the paper, and I think this was also a quite important paper because this was the first time that it was shown that you could inject venom, whole venom, into a mouse, wait a little bit, and then inject an antibody and save the mouse's life. This is much more difficult than pre-mixing and injecting. But this work also had limitations. We had gone about it focusing mostly on, you could say, one toxin at a time, as we see over here. If you do that, you would have to make an antivenom if you want to cover multiple species that has a lot of different antibodies. Ideally, you make broadly neutralizing antibodies so you can have a reduced mix that covers more snake species. So for this, we kept looking a little bit at the monocle cobra, but also looked at toxins from the black mamba and the forest cobra, where you can visually simply see that they have pretty much the same geometry. And then we used a method that my specialist Sheeran was the first to sort of report, at least in the toxinology field, where we did phage display but exchanged the toxin each round, so that we selected binders in the first round to one toxin, but then selected the subset that would cross-bind, and in the third round, the subset that would cross-bind even more. And Line used this technique to go through the library again and find antibodies that could fully protect against the main venom and also prolong life expectancy of mice challenged with king cobra and black mamba venom. And importantly, we also did these rescue assays and showed that these antibodies, they also worked in a setting that more closely mimics a snake bite. And here's the paper to those that are interested. Another important paper where I think we report the first therapeutically relevant antibody. As a small sidetrack, but because it's important later, I want to highlight some work done by my assistant professor Esperanza, where we thought instead of doing this cross-panning, where you actually need many more toxins and then they can be complicated to obtain, why not design a so-called consensus toxin that is an average of different toxins from different species. And Esperanza was ambitious. He didn't just choose 2 different scorpions that have different venoms. He even chose a spider. And they might, to humans, look a bit the same, but this would be kind of like choosing a human and E. coli and taking an average. They are quite far apart evolutionary. But they by some evolution, well, I wouldn't say coincidence, but there's probably an evolutionary reason behind it, but by coincidence, they have some toxins that are similar in some cases. And what Esperanza showed is that if you use these consensus antigens and pan against them, you can sometimes get antibodies that have uncompromised high affinity, but have that broadly neutralizing aspect that's important. So quite a nice technical paper here. So during all of this work, we also thought, my group and I, hard about what is the right antibody format. And we'd originally thought it would be human monoclonal antibodies, but we started looking more and more into so-called nanobodies. And nanobodies are these small antibody fragments that come from camelid species. They have pretty much all the same benefits as large, regular IgG antibodies, although their half-life is shorter. That's an issue. But on the other hand, they are much cheaper to manufacture in large scale. And as we've shown later, they can also penetrate deep tissue. Or others have shown they can penetrate deep tissue, but we've shown that they can also do that in case of snakebite and go out and neutralize toxins distally. The way you find these is very similar to how you find human monoclonal antibodies with phage display. The main difference is you have the benefit of being able to immunize a camelid from which you derive the library from, so you can use all the benefits of in vivo immunization and in vitro display to select super good antibodies, or in this case, nanobodies. And the first study I want to highlight here is my other assistant professor, Melissa, who basically did all of this on 2 different key toxin families from coral snakes, and showed that you can rescue mice that are challenged with whole venoms from coral snakes using just 2 nanobodies, which is quite elegant. And let's see, and this is the paper here. And I think to highlight this is where we went from having a therapeutically relevant antibody itself that is not an antivenom to actually having the first real antivenom that could probably be made into a product. It jumped. That's the paper. And the last paper in the discovery area that I want to highlight is my former PhD student Anna, who also used the consensus strategy to isolate broadly neutralizing and high affinity nanobodies. And importantly, in that work, we also crystallized the nanobody together with the toxin and could show why is it that they are broadly neutralizing. And I wouldn't say that it was particularly surprising. We had the hypothesis that if these nanobody mimic the natural receptor, that all these different toxins they bind, there's a good chance that they will be broadly neutralizing. And that is exactly what we saw in the crystal structures, that they neutralize the toxins via, you could say, a receptor mimicry mechanism. So to those of you who are as smart as my little daughter here, you will of course know that last year was the year of the snake in the Chinese calendar. Fun fact, in 2013, when I started doing antivenom research, at least on the desktop, it was also the year of the snake. But I want to also say it's actually still the year of the snake. It's good that we have this defense this week, because already next week I think it shifts to be the year of the horse. We can think a little bit about that, but because of that, I want to sort of at least touch upon one big study that we've done and published after the thesis was submitted. And I'm going back to all these elapids that we had already characterized. this year we showed that we could make a simple mixture of just 8 nanobodies that can neutralize 17 of these venoms, and I think it won't take us too much to extend it to the last green mamba here. And I would say this is really the culmination of all the methods and ideas that are in the Dr. Tech dissertation that we showed, led by Shirin, my specialist again, and my current PhD student, Nick, that we can't just do it with relatively simple coral snake venoms. We can actually do it across different genera. So that means that we start having something that can have continent-wide coverage, and that's, I think, important when you want to sort of go into the next translational steps. And then, as the last section on work that went into the thesis, I would like to share a few words on manufacturing. It's not something that we've done enough on yet, but it's a bit ongoing. When you want to make antibody mixtures, you can either do it in a parallelized manner where you have each monoclonal antibody made in a bioreactor and purified and then mix it into a cocktail. That gives you some control, but you also need to run many batches. There are technologies out there that allow you to co-cultivate different cell lines that each make different monoclonal antibodies so that you can actually make the mix in one batch. And my former co-supervised PhD student, Christina, she showed that it is actually possible to develop such cell lines and control with good agreement between theoretical calculations, what we actually saw in the bioreactors, that you can grow these oligoclonal cell lines and make oligoclonal mixtures of antibodies. And this came out, well, it's actually 2 years ago now. But the obvious thing, now that I've already presented something on nanobodies, is can you do it with nanobodies? Because nobody, to my knowledge, has ever made mixtures of nanobodies, or for that sake, any antibody fragment. And this is ongoing work, as you can see. It actually says submitted, but I think that's a lie. That was the aim. But my senior scientist, Anne, and senior scientist from our collaborators at NGenes, Patrick, have led a study where we are mixing cell lines that make different monoclonal nanobodies and grow them in one bioreactor. And you can see here that the important aspect is, I can tell you it works. The important thing is this number down here, where we've tried to do a techno-economic assessment of what does it actually cost to make a real antivenom, a recombinant antivenom product, because I think that's still one of the main critique points about the work here. Will it be low cost enough to actually reach the people in need? And I would argue that this level is hopefully acceptable for saving a life, no matter where you are in the world. And just for reference, the good antivenoms in Africa right now, they cost around $1,000 per treatment. So it is, if we can reach this number, which our early pilot scale studies indicate, then I think it's okay, even if we go a bit higher. Good. And then rounding up, just a few words on how I see the future. I think it's important to stress that I don't think next generation antivenoms will just be one thing. I think we will see a mix of modalities. I think we will see human monoclonal antibodies. I also think we will see nanobodies. And I also think there's a range of small molecule inhibitors that can either be used alone or in combination with these antibody formats. And that we will see products coming out in a more differentiated way than we've seen so far with plasma-based products. I've already kind of indicated the benefits of doing this. I think it's to keep it short. We can improve safety, efficacy, hopefully also lower cost, and thereby benefit all the snakebite victims that are currently not receiving the antivenom or treatments that they should. I think it's important to highlight that snakebite is a bit of a complex thing to manage because it's a medical emergency and there are many players. A nice recent paper from my mentor and great friend, Professor Gutierrez from Costa Rica, made this nice figure showing both that the community plays an important part. You need to be educated on what to do in case of snakebite. You need to know how to respond to it in terms of first aid. You need transportation means. Often, many, not all, but many snake bites happen in rural areas, and you need to have good healthcare centers, those places with access to good products. Oft you will also need to have more advanced tertiary settings, also with access to good products, and you need to have good follow-up on people. I think it's important to highlight that diagnostics, I think, has maybe been a little bit overlooked in the past. In my opinion, should play a bigger role. It is very hard to know what snake bit a patient, and therefore how should you treat them. Not just what antivenom should you give them, but also should you have ventilation equipment? Is it a neurotoxic bite? Should you have that ready? Should you have extra blood? Do you expect to need a surgeon to deal with complications from the bite? So I think to sort of round off, I think we need better in vitro models for studying snake toxins and for developing recombinant antivenoms, or at least next generation products. More work needs to go into low cost manufacture and establish also how regulatory pathways should look. I think we'll see hybrid products. I think we'll see diagnostics moving into the field. I think there's a business model that needs to be fixed right now. There's an issue with it not being profitable at all to go into, hardly profitable in most places. That's a problem if you want new innovation. And I think we will see a differentiated, you could say, portfolio of different types of both products and solutions coming out over the next decades. And you could say, as final conclusions, and the first one is those are mostly to my own field. I think there's been several decades where sporadic efforts on discovery and testing out hypotheses has been going on. I think we're reaching a point now where we need to consolidate efforts and focus more on translation, and not just keep saying, oh, you never know where the new big discovery comes. Now we actually need to take some choices and bring things forward towards the clinic. That being said, there should of course be a holistic approach and not put all eggs in one basket and focus on only one thing. But I think we do need to consolidate the field. I think it is mature. With that, I'd like to thank a few people. First of all my fantastic co-authors on all the papers behind my thesis, here in alphabetical order. Those of you that are quick will see there's exactly 101, which can sort of question my role. Beyond the co-authors, I of course also want to thank my fantastic team here at DTU, all my many collaborators from both companies and universities, and my fantastic funders that have really been generous of funding the work that went into this thesis. And with that, also thanks to all of you for your attention. And I'm supposed to highlight that if you want to register as an unofficial opponent, as Camilla said, you can write an email. You can maybe also just go over in person if you're here. And that brings us to the conclusion of my part. And there's now 15 minutes, 10 minutes. There's 10 minutes break. Thank you.
So at this point, I'm going to ask our first opponent, Professor Brian Fry, who is with us online from Australia, to take the first questions. Thank you very much.
Well, absolutely fantastic work, Andreas, as I'm sure you already know. So just have a few questions. It's kind of, because I have to probe these areas. So the first is on target selection and prioritization, going from your venomics to toxicovenomics area. Just first want to check, can you hear me okay?
I can hear you, yes, and I'm looking on you on a screen that is different from where the camera is.
All right, cool. All right, so this all goes, I guess, a bit into sort of the future aspects where with the African elapid venoms, they've got fairly streamlined venoms in regards to the three-finger toxins and the PLA2s, basically two toxin types dominating across the continent. But when you go into like with the vipers, where it might be much more complex and a lot more isoformic variation. So in that idea, sort of just speaking broadly, how does your systematic approach to venom target identification translate into a rational minimal set of toxin targets for these recombinant antivenoms? And in doing so, how would you account for toxin abundance, potency, and also synergism, which is an important aspect in itself when ranking these kinds of targets?
Super good question. That's not one question, Brian. That's a lot of questions. But let's see if I can take them one by one, and then remind me if I miss one. But I think you can use the same framework. And to just answer maybe one of the last questions, it's then just a question of how broad can you go with one antibody or one binder. So of course, I think with the phospholipases A2 that you mentioned, I would not expect that we can neutralize all of them with just one antibody. I think there will be subgroups, just like we actually see for the three-finger toxins, where you can't, at least we've not succeeded with making a binder that both binds long and short neurotoxins, despite that they have overlap in targets, receptor targets. So I think it's a question of just looking then at subfamilies that group together. I do think with the enzymatic toxins that I think it can be done with antibody. I mean, it can, because we know existing antivenoms work. We know that they can be neutralized. But this is perhaps also one of the places where I would question whether it might make more sense with a small molecule inhibitor, so that you use the substrate specificity, which might be conserved in some cases, at least more broadly. It's a little bit hard to say what's best. It's one thing is, of course, what neutralizes best, but what is also the easiest to manufacture. So I think, the simple answer is, I think you can use the same framework. You just might need to go down to subfamily level when you talk about broadly neutralizing antibodies. I think touching upon synergism, which is something I've also studied a bit, and at least an area that fascinates me a lot. You can look at it systematically, and I think the question here is, if you have two components that synergize, as you might see with taipan venom that I know you're very familiar with. In that case, they synergize simply by binding together. You could be lucky that simply just abrogating that binding, that you just need one binder to one of them, you can destroy it. In other cases where the synergism sort of happens on a more physiological level, where something happens to the capillaries, something else damages the coagulation system, I think you probably will need to neutralize both toxicities. Even if there is synergy, you might want to go against both. But with all this being said, I think at the end of the day, if you are to develop a new type of product, you need to take a decision on your target product profile, on how perfect is it going to be in terms of therapeutic efficacy, how perfect is it going to be on safety, how perfect is it going to be on manufacturing cost. You can't have all. If you want to make a cheap antivenom, it is likely not going to have as many components or as advanced components, and you will have to compromise on therapeutic properties. If you want to make the therapeutically best antivenom, you'll have to compromise on cost, meaning that you'll have to pay more. So, I mean, we are working with viper venoms. I think I maybe didn't include anything in my dissertation on it, because it's the next, you could say, phase we're looking at. Part of the reason I also chose not to work with metalloproteases and serum proteases is also that I can see small molecules having traction in that area, and there's no reason to sort of go head to head against that when it's pretty obvious to me that some of the small neurotoxins are extremely unlikely to be neutralizable with a small molecule for simple biophysical reasons, like there's just not enough possibility for two small molecules to interact in a sufficiently strong enough manner to neutralize, or it would at least involve such complex chemistry where you then transfer irreversible binding groups and things like that then it becomes a really complicated discovery effort. I think that hopefully answers most of what you asked about.
Yeah, very good answer, or answers, as the case may be. Obviously it comes down to the fine art of what is possible, what is doable, versus what is, like you said, what is optimal. So yeah, no, that was perfect. All right, so moving on to more sort of the engineering side of things. I was fascinated with the idea of the cross panning versus consensus toxin strategies, and just wanted to have you go into that a little bit more broadly, where thinking of it, sort of, under what biological or methodological conditions would you expect one strategy or the other to outperform the other one? And, sort of what key failure modes are you kind of incorporating into that? Just like broad binding that doesn't necessarily yield in vivo protection.
Yeah, good question. So also just for sort of some fun, originally I thought the consensus idea was a terrible idea, but I had to submit a grant application that had to be different from some previous work. So I had to come up with something, but I thought this is unlikely gonna work, but let's try. So I don't think if I turn your question around, I don't think it's easy to predict which strategy is best. I think you can maybe to an extent say that if you have enough structural knowledge and kind of know where the neutralizing epitope might be on a toxin, or if you know that there are some domains that are more important than others, then I think maybe the consensus strategy is more likely to be successful, regardless of whether it's better or worse than the cross-panning, but that gives it better chances because it is, you could say, a strategy that involves or gives you the possibility of having more control on your design. If you have no idea on where to bind, maybe the cross-panning strategy is preferable. I say maybe because it is the real targets you're panning against. So what you find that works will likely also then work in reality. So I wouldn't say one strategy is better than the other. I think it's just two complementary strategies. You can be lucky that in some cases, which we've been with the consensus toxins with the consensus neurotoxins, that it's actually worked surprisingly well. But I think we could just as well see cases where it's the opposite. I think it's important to say when you design consensus toxins, which is something we don't describe that much in detail in our papers, is you can sort of design a consensus toxin in three ways, roughly speaking. You can take all your proteins, align their amino acid sequence, and then just take the average and let the computer, just like when you blast something, just let the computer select the average. That's the default version. That's going to be an average toxin that is neither the best at getting recognized nor the best to use as an immunogen. If you think a little bit about just geometry and you align these different features and align these different amino acids and you start thinking about what features do you want to sort of introduce, then in cases where there's, let's say, conflict, where there's disagreement on what amino acid should be chosen, then it's a little bit like a lock and key. If you want something to be recognized by as many antibodies as possible, which is what you might want if you want to diagnose patient sera, you should choose, let's say, the minimal design. Instead of having a big consensus toxin, choose the smallest one because it's going to fit into the glove of most antibodies. Vice versa, if you actually want to induce broadly neutralizing antibodies, you want to induce antibodies that have as big gloves as possible, so you make the maximum consensus design. And I think this is one of the smart things that my assistant professor Esperanza led, was this design aspect, or you call it algorithm, but it's just, a human brain algorithm, that you actively take some design choices on the physicochemical properties and the size of the amino acids when you design your consensus antigen. And I think without doing that, the consensus strategy is not particularly good. But it's an easy-ish thing to do. And I think when you do it, that's also the reason why we saw the amount of success that we saw, both with human monoclonal antibodies and nanobodies in our discovery work.
Another great answer. Not that I expected anything else. I know you're going to crush all of these, but you've got to go through them. So, I was fascinated by one aspect of, the ADET, when the antibody engineering basically backfired, and the fascinating report of, the antibody-dependent enhancement of toxicity that, where you've pointed to a YTE mutation as being responsible for the half-life extension. What mechanisms do you consider most plausible, and what preclinical study design would you implement to systematically de-risk FC engineering for antitoxins, including, like what muzzle choice of pre-incubation or rescue versus challenge?
Yeah. Good question, Brian. I'm just putting up some data just so the audience and also you can just see what we're referring to. We discovered in my former both PhD and postdocs, Christopher Sørensen's work, that depending on, at least in some cases, if you bind a toxin, and especially apparently if you engineer antibodies to have a longer half-life, they don't neutralize the toxin, they actually enhance its toxicity. They also likely change the toxicity. And our speculation, which is based a little bit on findings from the 90s on mushroom toxins, is that what? Why does it? Sorry, I need to. What we think could happen, because what we saw was that when we mix toxin and antibody before we inject it and then inject it, then it works perfectly. There's perfect neutralization, and our expectation is that it's because larger complexes are formed and they're just deposited somewhere subcutaneously in the mice and don't really do anything. But when we inject toxins first and then inject the antibodies IV, what we expect happens is the toxins, they leak out into the bloodstream, latch onto an antibody and then stay longer in circulation, and we can at least conclude that somehow the body is exposed more to the toxins when this happens. The histology pictures that we see here are from some of the mice where we did this, and what we saw was that the so-called Bowman's capsules that are pointed out with arrows, they were destroyed when the toxin was injected first and then the antibody was injected later. And what we think happens is that the toxins, when you just inject them, they might disperse around. They cause a lot of muscle damage, but it's spread a little bit, diluted all over the body. But when they're onto an antibody, they won't be spread out as much, and they'll at some point end up at least passing through the kidneys. And exactly what happens there, we don't know, but somehow the toxins are either released from the antibodies due to microenvironmental changes, maybe, or simply have some inherent affinity, at least in the form they are bound to the antibody, and get stuck and destroy this capsule. And kidney damage is of course a very severe, or nephrotoxicity is quite a toxic effect, also more toxic in terms of lethality than muscle damage is alone. So you could say this is our speculation on what happens. Then when you talk about antibody engineering, what could you do to prevent it? We are looking more into it. My assistant, Professor Melissa, has a grant where she's studying this in more detail to try to figure out what is the mechanism and how can we avoid it. One hypothesis could be using a smaller fragment, such as a nanobody, that when it's bound to many of the toxins will be small enough to actually pass through the kidneys when it's below the glomerular threshold. Maybe that's a strategy to simply just neutralize it and get it peed out, and it won't stay around in the kidneys long enough to do enough damage. Maybe that's a hypothesis. Of course, it could be that we need to bind a different epitope. And the last thing is actually in the binding interaction between the toxin and the antibody. And one thing we write it in the paper, but we don't spend too much time on it, is that the antibodies we selected, they were actually selected using pH elution, meaning that they let go of the toxin when pH changes. It could be, and I tried to figure it out, but what I can read, pH is not that different in the kidneys as I would have expected. It's a little bit different, but it could be that somehow the microenvironment favors that the toxin is released there, but not in blood. So, it could be that instead of having pH responsive antibodies, which these might be, the thing is we can't actually show, at least we didn't show that whether or not they were, because they bound so strongly when we test them in vitro that you can't really see if they bind stronger or less, because they just bind too strong. But under physiological conditions, it might actually be that they respond to changes in the microenvironment and let go. And then you can say we did some changes to the FC region of the antibody that made it worse. And I think, in essence, that if an antibody binds and prolongs the exposure of the toxin, wherever that is in the body, that if you engineer to have an even longer half-life, you'll just exacerbate that problem. We also removed effector function on the antibodies, but I don't think that influenced what we saw here. So I do think this is probably not a common phenomenon, but it's certainly a phenomenon that you should be on the lookout for. And one of the interesting things that we'd like to study, Melissa and I, is whether this actually happens with existing antivenoms. Not that all of the antibodies might do this, but could there be a subset of antibodies in existing antivenoms that actually work against the rest of the antivenom? I wouldn't rule it out. And if that's true, that gives an even stronger case against continuing to use plasma-derived products. which could simply be avoided by using properly designed, well-selected monoclonals, where you've avoided this feature. And I mean, it is well known that local tissue damage and myotoxicity and things like that, it's not neutralized very well with existing antivenoms. And I think the main standing theory is that the antivenom doesn't reach out in distal tissue fast enough. And that's probably part of the answer. And I think it's maybe also the largest part of the answer. But I would speculate, maybe there's even a subset of antibodies that's working against us. It's at least a hypothesis that's worth investigating, because if it's true, it's quite important for future design.
Oh, absolutely. If that's true, that's opening a big, can of worms. Absolutely, intriguing answer. I like it. And I look forward to seeing what you guys come up with on that. All right. So moving on then from discovery to GMP reality, when talking about your single batch oligoclonals, which is, obviously, the desired outcome. So because, as you presented, it has a good cost competitive manufacturing aspect to it. But how would you address the dominant technical risks, including clone competition, drift in relative expression over time, or downstream purification issues? And with these, obviously these variations that can, change over time, how would you define and control the mixture identity so that it's stable in order to satisfy regulators?
Good question, Brian, and also slightly out of my true comfort zone, to be honest. I mean, of course, you can solve it by not doing oligoclonal mixtures if you have components that you want. And I think, the reason I say that is I've been a big proponent for oligoclonal mixtures, but seeing that our components also work against some of the Asian snakes, I would maybe go a little bit back and say maybe some of the components should be made individually, because then you can use them for different products, one for South Asia, one for Sub-Saharan Africa, and maybe other places. That being said, if you want to go for oligoclonal mixtures, there's what can be done to some extent is, of course, that you ensure that you work with cell lines that are well described, well understood, well studied, where the genes have been inserted into these landing platforms that we've used in the CHO cell examples, and that you simply have more control by designing and characterizing and studying your cell lines well. That's what has been done in some companies like Symphogen, and that gives I think, sufficient level of control to satisfy some regulators. There are different companies out there that are developing, let's say, different technologies for making cell lines addicted to the product they make, meaning that if they stop making it, then they get abstinences. And this favors the high producers. You could then, of course, question, could they then drift and make too much of something? Maybe that could happen. But of course, if this turns out to be an issue that you can't just design your cell lines well enough and use those, there are methods to implement from other parts of the sort of biomanufacturing industry, also with making auxotroph cell lines that depend on a nutrient or a cofactor. I mean, you can't use antibiotics anymore, but having certain, you could call it selection markers, they just can't be antibiotics anymore, but having selection markers right next to the gene of interest, your antibody, can help with the control over, you could say, the growth of your cell line. So I would say there are ways to, I would say, solve it, or at least mitigate it dramatically out there.
All right, I'm fine with that. All right, so moving on to how you go about measuring cross-reactivity in a way that actually predicts efficacy. Where in your various papers you've discussed multiple analytical techniques and approaches in regards to cross-reactivity assessment, epitope profiling, antivenomics, style pull down, mass spec, et cetera. So how would you integrate these data with functional assays to avoid false confidence from binding only readouts, which is especially for conformational epitopes and toxin families with diverse mechanisms?
Yeah. Good question, Brian. And I have an example just with neurotoxins, because I fully agree that it's important not just to look at binding. And a lot of researchers have just looked at ELISA data or things similar to ELISA where they claim that something is cross-reactive, and one thing is that binding might not translate into neutralization, but these types of data are both dependent on how much toxin was there, how much toxin actually absorbs to an ELISA plate. Some absorb really well, some don't, some absorb in the wrong orientation. So I would say such data it's not useless, but it's close to useless, at least on its own. So I'm a big proponent of doing early in vitro functional characterization of cross-reactivity. And I mean, I showed here we use a nice patch clamp assay that the company Sophion has made, and I know your colleague Glenn has I believe he has one in his lab and is studying this with spider toxins and other things. And I honestly don't think you can truly claim that something is properly cross-reactive or cross-neutralizing unless you test it with some kind of functional output. And then bearing in mind that even that functional output from in vitro might not be what you see in vivo. But again, these things are also a question of how many resources do you spend on different things, when do you spend them. Ideally, you test, I mean, from an engineer's point of view, you ideally test all of this in humans. From a more ethical person's point of view, you move to animals or in vitro. But ultimately, it's a question of how much effort do you spend on studying neutralization, how many venoms do you include, how many toxins, because if you keep on just studying cross-reactivity, well, then you also never leave the station, right? You're stuck there. But like we've done with patch clamp, where we've the limitation there is that we have to isolate the toxin, so that's the bottleneck. Because if you use the whole venom, often there are cytotoxic components that kill your cells, so they don't make these nice recordings from the ion channel resistance. So even though these things sound nice that they're in vitro, they're high throughput, which they can also be in some of the enzymatic cases, which I know your lab has studied with blood coagulation. The issue is, of course, that given that snake venoms are quite complex and have multiple different components, that studying a whole venom has its limitations in vitro, because you might have cytotoxins that destroys the cells if you're doing cellular experiments. If you're studying enzymes, you can have other enzymes in your venom that actually start chopping up the toxins themselves, that either autocatalyze or catalyze catalyze the degradation of the neighbor. And I'm going to promote Melissa a little bit again in some of her work, where we are looking at some of these aspects with snake venom metalloproteases, where we see that, I mean, we see some nanobodies and antibodies neutralized. We see some they don't do anything. And it turns out maybe some of them without FC regions actually might enhance toxicity a bit, and maybe even more than a bit. So yeah, it's always with some risk when you study things as isolated components, but it's also a risk to study them when there's synergy happening and counter effects and so on. And ultimately, what we keep on discussing at our meetings is these things we see in vitro. Do they actually happen in vivo? Are they meaningful? I mean, my PhD student Christopher, who did his bachelor with you, he also found some antibiotics that enhance enzymatic activities of some venoms, and in reality, it probably has zero meaningful effect in an animal or human. But in vitro, some of these antibiotics turned out to enhance enzymatic enzyme-driven toxicities.
It's a very comprehensive answer. Thank you for that. So last but not least, looking at sort of ahead, what is in regards to your strategic roadmap for these next generation antivenoms in terms of clinical translation, regulatory path, and real world access? So thinking about the constraints about, cost, distribution logistics, and safety limitations of the existing antivenoms, these heterologous plasma products, what is your proposed near-term most deployable solution? Are you thinking fully recombinant oligoclonals? Are you thinking hybrid where you've got existing antivenoms fortified with some of your defined monoclonals? or alternative scaffolds? Sort of what's your strategic roadmap in this regard?
Good question, Brian. And I think the answer is a little bit that it depends on whether it's something that I'm trying to push out or something where an existing manufacturer might want to take a next step. And if we separate you could say, world, or at least the sort of translational word, into sort of existing big companies that want to do innovation, do something new, and small companies that want to do something new. Bigger companies can allow themselves to do just a little bit more incremental innovation. And I could imagine that existing manufacturers could take a particularly good nanobody or antibody or small molecule and mix it into their existing product. If they know that their product doesn't neutralize a specific toxicity very well, the relatively easy solution is then to add a broadly neutralizing antibody that then does the job. This could be the case for many neurotoxins, I think. But I think if you want to spin something out, if you imagine making a small company or something like that, I think you would have to be bolder and more visionary. And if you do something new, you also don't want to both have to establish biomanufacturing and a horse farm. You kind of have to choose. So I think I would say if you want to sort of push something from the bottom up, it would be fully recombinant mixtures of either nanobodies or fully human monoclonals, maybe supplemented with small molecules. But given that the small molecules are being championed by 2 or 3 companies already, they could also, in a clinical setting, work as adjunct therapy, that you simply administer 2 products side by side. It's not unheard of that a patient receives more than 1 drug at a hospital. I think to get there, it's important with especially the newer formats, such as nanobodies, that we get some kind of data from a large animal model, such as a sheep or pig. We have good data in mice, but the pharmacokinetics are very different in mice and man. And if I remember correctly, you some time ago also showed or argued that what you see from many of the hemotoxic snake bites in mice is a completely different scenario than what you see in humans, whereas in mice get blood clots, whereas humans get consumption coagulopathy because of the different volumes. And I think a little bit, regardless of what venom it is, that we need to understand how well do the different new types of components function in a species or an organism that's the same size as a human. It will be quite informative for designing target product profiles, deciding on ratios between components in such profiles, and ultimately understanding what initial doses, or if there has to be follow-up doses, how you design such a dose regimen. But as I also had in my last slides, I think we will see a mix of different types of products. I think we will see some standalone small molecules come out that will supplement existing therapy. In some cases, they might also completely replace existing therapy, especially in cases where existing therapy is not deployed. I think currently it's 2% of victims in sub-Saharan Africa that need antivenom that actually receive it. So you could say anything that comes out there that's safe and has some efficacy is needed. And even if it's not a perfect product, it's a standalone small molecule, I bet that people that know that these things have benefits would use them if they are affordable. So I think it will be. I think there are different paths. I think there are different players. There's maybe going to be some smaller companies championing some slightly more bold ideas. I would hope existing manufacturers would both champion, let's say, the bolder ideas with completely new types of products, or at least start using some of the good components as supplements to fortify their existing antivenoms. I think it makes so much sense. I think the big question here is, how will regulators view that? Because if you need to go through a very lengthy, costly process every time you supplement your antivenom with something, it will be a hindrance, a barrier for bringing these things out. But there might be cases where adding a few components will dramatically improve your antivenom, where it's worth doing, maybe. But it's a economic question really more than a scientific one.
Well, that's it from my side. Brilliant answers. I'm satisfied.
Thank you, Brian.
So thank you very much, Professor Brian Fry. And With this, we go to our next official opponent, Professor Michael Hust. You are welcome to come to the podium. Sit down if you want. Please proceed.
Yeah, but then they can't see me. I'm too short.
So, now we're coming from the one guy with the black shirt to the other guy with the black shirt. So. First I wanted to ask also some. Then, oh yes. Yeah. Whoops. First, I wanted to ask also some venom-specific question I'm interested in, or very interested in. Looking on figure 7 of your thesis, perhaps you have it. It's the image generated with your online tool, showing the compositions of in the venoms of the different toxins.
Mm-hmm.
Perhaps. And there you have, oh. Huh? Okay. Okay. There you have the Naya Kautia, well, however it will be pronounced. And the composition of the different toxins in the venom is completely different in Thailand, Vietnam, Malaysia, and so on. I asked me, what is the reason for this? Is there a genetic difference in these between these single species, or is it a physiological? Okay. Or is it a physiological adaptation to the environment or to different species they hunt? What is the reason for this?
Yeah, good question, and probably something we could spend a lot of time discussing, because it is, as you say, interesting to see that the same species, or at least what we have designated being the same species, has quite dramatic venom variation. And it depends on the geography what the reasons are for this. And in some of the cases that are simple to explain, it could be that a mountain range has split, or at least the species has been split across a mountain range, and where over thousands and thousands, maybe even longer, millions of years, that one population has drifted towards a slightly different type of prey, because one side is more dry and maybe has more lizards, whereas the other side more wet and has a different type of prey. And snakes with a venom composition that's optimized towards prey in their area will have a evolutionary benefit. And maybe it's important to say that the hypothesis is that at least that the majority of the selection pressure on snake venoms and how they evolve is prey, what prey they eat. It's not so much used for defense. There are some exceptions with spitting cobras and other things, but it's very much how effective do they not necessarily kill prey, but immobilize them so they can eat them.
Were these species sequenced to see if there are any difference in the promoters for these toxins?
To my knowledge, it's only around somewhere between 20 and 30 snake genomes that have been sequenced, and we recently published the only true viper genome where we sequenced one individual. It has the deepest, the broadest coverage, and we're the ones that went deepest with his genome. And just doing it with one snake, you can't really say much, because how would it have looked if we took another one? But I think the speculation is that just like any other species, including humans, that when you spread over geographies and you let time pass for a sufficiently long time, you will see a drift in genetics, whether it be that, I mean, in snake venoms, whether genes they get duplicated. That has happened a lot in snake venoms. The toxin genes have been duplicated. Originally, they were, you could say, duplicated and neo-functionalized from proteins that you could say animals have in their body. I mean, I think some of the enzymatic toxins were probably neo-functionalized from digestive enzymes. And if it gives the snake species a big enough evolutionary benefit, they will then the venom or the snakes that have the venoms that are better adapted for the environment will have a natural advantage. We actually also see the opposite. And I think it's the Burmese python was the first snake genome that was sequenced. The Burmese.
Okay.
And there, surprisingly, it was found that this snake actually has venom genes, but it doesn't use them. It uses constriction, strangulation, to immobilize and kill its prey. And it's of course hard to know why did it happen. And I don't think evolution doesn't have an intention of doing anything, but the hypothesis is that this snake grew to a size where it was just more effective to strangle its prey rather than have to make venom and inject and so on. And there's a case with the marbled sea snake. Sea snakes are generally neurotoxic, but the marbled sea snake has some genes for neurotoxicity, but some of them have premature stop codon, so it actually expresses toxins that are useless.
Coming from gene duplication, and then by coincidence,
It has probably, let's say, its ancestor has probably been neurotoxic, but this snake likely changed its diet. It at least eats mostly fish eggs. Other sea snakes, they eat fish, and fish, they can not just swim fast, but they can swim in 3D. Terrestrial snakes, they actually only operate in 2D, you could say. Sea snakes operate in 3D, so you could say it's really important to have a fast-acting neurotoxic venom to, immobilize the fish so you can eat them. But if you shift your diet to fish eggs, they're pretty immobile already. So it doesn't mean that this has forced the venom to stop having its toxic function by having these premature stop codon. But that just happened. And somehow maybe that was a small benefit, or at least nothing happened against those snakes that were no longer particularly venomous. So they have thrived and are still making. So they are probably midway in their evolutionary trajectory with this tox, because the best of these, the most adapted snakes, they would actually stop making this unfunctional toxin because it has a metabolic cost to make it. Probably not so much. They're probably living in nice Australian waters where it's warm and there's plenty of fish eggs. But if times get rough, you would probably see.
Get rid of it and then have an invention. Coming to a very Danish question. So, I have to look at the English word. The Danish word is the Hougaard, the European adder. This is the only really toxic snake here in Denmark, also in northern Germany. In Norway, I was nearly stepping on one. My wife said, "Please stop."
Yeah.
Otherwise, perhaps she started to bite me. But you also have something against this in Danish Hougaard?
We have not worked on that simply because...
So you're looking far away, but you're not looking close to your own country.
Yeah, I think just as a fun fact, it's not a hug worm. Don't hug it. It's it's also not a hygge worm, but no. But you could say it. I think it's a common belief, probably also in Germany and Western Europe, that getting bitten by one of these European vipers, it's just like getting stung by a wasp.
It's not very...
That's not true. Well, it's true if it delivers a dry bite, where it can, still give rise to an anaphylactic shock, just like a wasp sting. But if it actually injects venom, its venom is potent. The thing is, it's just not a very big snake, and it doesn't have a lot of venom. So in most cases, because it both delivers a lot of dry bites, it cannot eat a human. Even if it does inject venom, we can still just chop off its head. So it would rather just chase us away and save the venom for someone else. That's at least the theory. But a mix between that, it doesn't really cause that much damage, although those that are bitten by it can suffer more than you would expect. A former student got an instant interest in snakebite and antivenom after he was bitten by a Swedish viper. And what happened to him was that his leg, it swelled to double size, and his foot was 3 times the size, and he spent, if I remember correctly, something like 5 days in ICU. And it took him 3 months before he could walk again. Okay. So of course he didn't die, and he's, I think, fine now. I'm pretty sure he's fine now, but that's also not so nice, right? You'd prefer not to have to spend 3 months at
That's right.
in rehabilitation. So, but nevertheless, you could say that we haven't focused on the European viper simply because if you want to actually get some of these products out and help people, I think you need to focus on a mix between where the largest medical need is. At least the medical need has to be large enough, which I would question in Europe, and also you could say where there's an absence of good products, because I wouldn't say that the antivenoms we have available in Europe are fantastic or perfect, but they are better than some of the bad ones. They're definitely better than no antivenom, which is the reality for some victims.
Regarding bringing antibodies to the patient later. Normally, your patients in Africa, Asia, they are in remote areas. So you have the challenge to bring the antivenom as soon as possible to the right site. Are you working also on lyophilization? So to keep it so that that you can keep it also in remote areas for a long time, and you don't need a cold chain. And
I would say we're not working on lyophilization, but we sometimes use it to freeze dry our antibodies when we ship them to partners just to do animal studies. And it's I would say it's already lyophilized antivenoms based on plasma-derived antibodies. They exist. It can be done. The benefit is, of course, that you have they're more stable when they're on the shelf. The downside is that lyophilization, it adds extra cost and complexity manufacturing. And if it's done suboptimally, it can actually take quite some time to reconstitute a lyophilized product. If it's done right, it doesn't take so long, but it does.
how much time does it?
Well, I've heard examples where it takes several hours to reconstitute, and that's a problem. Of course, if it only takes some minutes, then it's maybe fine, but it still adds the complexity that you then need to deliver saline.
Also on the formulation, so.
It certainly does. I mean, I'm not an expert on lyophilization, but what people say, if it takes several hours, then there's room for optimization. But it does then require that you deliver saline water with your antivenom, and that someone, a pharmacist or some person like that, would actually stand there and reconstitute it, which is just an extra step that's inconvenient when you are in a.
Perhaps it's good to have it, to store it for more time, having more shelf life. And
I think the question is, it's of course one strategy. I could imagine that in some remote areas, that not just for antivenoms, but let's say emergency medicine in more general, that you would store them regionally, but slightly centrally, and then simply have them delivered by drones. The challenge in many of these rural areas is really that it takes hours with a off-road vehicle to get through very bad roads, but I think this is where drone technology can deliver things much faster in that last few miles, and that means that you could have maybe.
you will go for drones instead of lyophilization.
Well, I wouldn't say that I would go for drones, but I see tests being run in different remote areas where delivery of essential products, essential medicines, are done by drones. And I mean, yes, if you need to keep something on the shelf, which might be the case in a clinic where you don't know if you see a snakebite patient tomorrow or it takes 12 months. But if you centralize it a little bit, you can probably average it out so you won't have too many products that expire and are wasted, that you could have them ready to go and deliver them quickly. It's
Now, keeping the point, bring it to the patient. I think, in my opinion, the real challenge for all of these projects, also many projects where I'm working on, is not making a good potential drug, is finding a right business model.
Yeah.
And this is, I think, the challenge. Well, and I think this is the challenge here. We have made antibodies against diphtheria, and there was a diphtheria outbreak 2.5 years ago in Western Africa. I had a call from the WHO and then the Clinician Without Borders and said, "Are you ready? Antibodies ready? Do you have a GMP produced? We can directly ship it to Western Africa because the horse cellar is running out." I said, no, because there's no business model to bring this to a patient, despite the fact that you have a lot of potential patients there. And there's another group in the US, Mass Biologics, they have the same problem. They have the same problem. So what kind of business model do you have to develop? Is it more public-private partnership like for the malaria vaccine? So
it's a good question, and
more challenging than science. Sorry,
no, but I think you're right. I mean, early on when I set out to do this, I also certainly believed that of course you can make products, but I share your let's say, fear or skepticism about can you actually get these things out. And as I also highlighted in one of my last slides, the business model is maybe broken in this area, because it's, hopefully at least for people, it's a one-time use only drug, but it's mostly needed in not just amongst poor people, but poor people in poor countries, to be frank. So either there needs to be, you could say, commitment from governments in those countries. I think we cannot rely on philanthropy, at least not fully. I think we do need backing from local governments that can see that this is worth, you could call it, investing in. And the good thing is that in some area, many areas, and it's been calculated that it's actually a benefit economically to deliver existing antivenoms to snakebite patients, because snakebite patients are typically young, either children or adults, working or playing in the fields, and if they die, that's one thing, but if they are amputated, they won't be productive. It's a financial burden for the families. Yeah. So at least in some areas, I hope that there will be backing to bring these products out and deploy them. I mean, at least we do have anti-venom today, and it is based mostly on publicly owned serum institutes in kind of all of the world except the US, and a little bit mixed in Europe, and Australia is also kind of hybrid. So you could say, at least in the past, governments have chosen, or ministries of health have chosen, this is a priority area. We are funding serum institutes because it's needed. Then it's a mix between how good quality of life do you want your people to have in a country, plus what is financially beneficial for a country. So I hope that in some areas that it will be, you could say, public funding that goes in and makes these public-private partnerships that you mentioned, and commit to long-term contracts that allow you to deliver things at a reasonably low cost, because you know you have a very long contract, so you'll have many orders in the future.
Main job would be to contact the health authorities, and
that's part of it, yes. And maybe philanthropic organizations such as Gavi and some other also like Welcome, that has funded a lot of the work, could play a role here, at least to get it started. But to keep it sustainable, there needs to be long-term commitment. I would also like to highlight a completely opposite business model, being the insurance one that we know from the US. And one could question whether in some cases this might be the right model, because one kind of benefit that snakebite has is that everyone fears it. Everyone fears it. Yeah. But it doesn't actually, if you look at an entire country, doesn't happen to that many. Yes, it's a lot of people, but if you take the areas where it happens the most, it's 1 out of 2,000 people. That's a lot that die from a snakebite. It's some particular valleys in Nigeria. That's a lot, but still, there's 1,999 that are not bitten and killed by snakes. But in an insurance model, if you priced it in a lower level so that you were ensured that if you have this insurance, you will get antivenom, then you don't need to charge so much per person, but you just have to guarantee that that one person, whoever it is, gets that antivenom delivered. So I'm not a proponent generally of commercializing healthcare and insurance. I am a big proponent of the system we have in Germany and Denmark of universal healthcare. But I mean, if the result is that no antivenom reaches patients, and a different model could actually bring antivenom to at least some patients, then that is the lesser evil.
Another point. You had a big discussion also with Brian on the model on the production system to make, when you make a mixture of antibodies, produce it separately. To my knowledge, this is also a regulatory nightmare finally, because of QC and everything, because you have to regulate this differently. And let's say the mixed approach, I will call it also Symphogen approach, because Symphogen was working with for many years. What's about bispecifics, quad-specifics, other molecules? Yeah. So why not going this way?
So I mean, we are testing out if we can combine a few, let's say, nanobodies into one construct to reduce manufacturing complexity. But ultimately, when you need to neutralize a snake bite, it's a numbers game. You have X moles of toxin that weigh X milligrams or grams, and you kind of need whatever ratio, but the same amount of antitoxins. Whether you then take these antitoxins and put them in a long daisy chain, you still need to make a gram of nanobodies, whether it's one component or a mix. And the more complicated you make your constructs, typically yields in your manufacturing process will go down.
I had more in mind
Yeah.
the more classical antibodies where you have one arm against
Yeah.
one, the other arm against the other target with a normal FC part, and perhaps at the CH3 additional single domains with which you can fuse, and you can make up to 4
Yeah. No, but
specificities in it. And here the drop in the yield is not so dramatically.
it all depends on that, right? So if the yield is still good, then that is a way to go. If you then need more than 4 components, you'll still have to mix, but it could at least maybe reduce manufacturing complexity. The thing that's important, this is also why I argue that we need data from a large animal, is that when you take a nanobody and inject it in a human, this is known from research done in Belgium, it is well known that nanobodies can penetrate more fast into deep tissue, such as tumors, which there's some nice work in Brussels that have been done. We see the same with nanobodies in snakebite, that they're small enough to actually penetrate out into deep tissue and neutralize toxins faster. We can actually better mitigate dermonecrosis, as an example, using nanobodies. And if you start sticking them together, you probably lose that. You lose that. Then you could argue, should you have some nanobodies and some larger? But then again, you then complicate your product even more.
Okay.
So I think, just to conclude, I think it's relevant to look into, but we need more data from a large animal to really take more informed decisions on what is the right antibody format.
Some nasty questions. So last year there was this paper from the Glenville group.
Mm-hmm.
Yes, and for me it was very impressive. You use mainly naive libraries for human antibodies, then you use also immune libraries for the VHH. They used an immunized human. For explanation, it's a person who was bitten by his own will for many hundred times by different snakes. So he has made your cross-reactivity screening directly in vivo.
Yep.
So what do you think about this approach? Finally, they use 2 antibodies and a commercial inhibitor.
Yeah.
This varadiplip or whatever.
Varespodip,
Yeah. Mm. What do you think about
Well,
it?
I mean,
compare it to what you have done.
It was no doubt that it's good work, and one can question, should you promote research that is like this or not? But if you take all those questions out and just look at the technical aspects and well, you could say it's the same we do in a llama, right? We immunize a llama and then we do phage display. They did kind of the same. Not they didn't actively immunize, but he was immunized. And then now I forgot if it was actually yeast display. But they used a display technique, if I remember. I think so. But
was it a combination of both first phage then yeast?
I'm in doubt. But they definitely took out and made a library and screened that library in vitro. They found a super good human monoclonal antibody against long neurotoxin, and then I would say they found an antibody against short chain neurotoxin. I would not say a super good one. If I remember correctly, the KD was 34 or something.
Was not good.
It's at least in our hands not good enough to go as much down in doses.
But there are combinations they made in vivo test with the venoms of, I think, up to 20 or 30 different
The concept that they showed, which is
we had something perfectly.
it is basically the same here, right? Which even this figure is from 2020, where we suggest that you could do this. Yeah. We didn't do it, but we suggested it. The concept I like. I like that it's a good sort of proof of principle article that shows, regardless of whether the antibodies come from, that you can mix antibodies, you can mix a small molecule into it. And this is the first time that Glanville and his group, the first time anyone did actually do that hybrid experiment. The rest of us have just said, maybe you can do it. So that's nice. I would then say when you take species from around the world, it looks like it is very broadly neutralizing. But I would argue that the venoms they used were cherry-picked. I would also argue that the doses they used, they kept it to a point where you would just see the influence of these two three-finger toxins and the phospholipases. So had you gone up in doses, some of the snake venoms would have caused more trouble. And you don't want a product that works against one snake in one region, but then the rest in that region. It's more important to have a product that actually covers the region you're in. That's a product. The other thing is a principle, a concept, but it's not really a product. So I think that's the main difference between, you could say, our later work, both what I highlighted from Melissa, but also our later paper that was led by Nick and Chirin, that we said we're not just we finished doing these concepts where we make a broadly neutralizing antibodies against toxins. We actually want to say, how do you make a mix that covers these species? Because ultimately, that's how a clinician will decide on what antivenom to choose. It has to cover the species in the area, and not one species that you might come across here, one in North America, maybe one in South America. You want your drug, whatever it is, to be robust and have clear indications of whether you can use it. So good paper in terms of scientific principles, in terms of translational value, not as much, and I wouldn't necessarily accuse the paper itself because they do list these limitations, but a lot of the hype that was said in the media where universal antivenom was almost there and so on, because that I disagreed with.
Up in the media also because of the donor. The story of the donor was a main, let's say, selling argument for this publication. Coming to something different. So the AI paper of Timothy Jenkins, and you. This was very impressive because I was always skeptical about making neutralizing binders. I'm often getting a publication or manuscript, I would say, mainly from the Iran, regarding as a reviewer or as an editor, and said, we have a new therapeutic, antibody, biological, vaccine, whatever, made it ready, it only has to be produced. And then they show some AI stuff, and that's it. No validation at all, not at no level. So I always reject it. And therefore, for me, this, your paper, or Timothy and your paper is very impressive because you made the models, you produced it, you showed binding, you showed neutralization in vitro and also in vivo. Yeah. What I would like to learn is, I did not fully understand what was the basis, did you use an original scaffold protein and then, or is it completely random? Or this is what I did not get. So I would like to.
Good question. So you could say again, I'd also like to highlight super good paper, but it's more the concept and the principles that we show. Also that it works, right? But because that's a big part of demonstrating the principles. But again, here we the translational value is less than our more later paper that came out. But as you say, there are many people that play around with computer algorithms, AI, and claim that things work well. And at least Tim has told me this was the first time that anyone had designed a binder and shown in an animal that it actually worked. Everyone else had done maybe some in vitro stuff and so on, but we were the first to show that it worked. And then it happened to be snakebite. That
and these binder, what does it depend on? Is it complete random sequences, or is it a scaffold?
Well, I'm not an expert on how the algorithms are designed, because there are some design principles exactly how it's encoded, and I cannot answer. But there's some motifs they avoid because they are immunogenic motifs. There will be some sequence improvement in terms of stability and expressibility, but I think in terms of what ends up binding, it is sort of random. There are some rules, not completely rational, at least.
So it could be also immunogenic later.
Well, that's the fear. That's what they fear. That people fear that they either are immunogenic, that they aggregate, and cause immunogenicity via that, or simply malfunction. I don't think anyone really fears expression levels, but sometimes it's brought up that you don't know. And then I would say then there's just an inherent fear from big pharma probably that say this is not even a scaffold. A mini binder, one mini binder is a completely different protein than a different mini binder. Whereas an antibody, it's still an antibody. You have a scaffold with smaller differences. So there's an inherent risk. It's
easier with an antibody because they know it.
We know what to look for. And I think there's also just an inherent fear that we might not know what we're looking for. Just like discussed with Brian with our antibody-dependent enhancement of toxicity, that was not something we expected.
No,
Similar type of things, maybe not enhancement of toxicity, but something unexpected might a completely new scaffold do that. That could be a fear. I think just because you also asked how did it work, and then I think the cool thing we did here was that we used primarily two main algorithms, AlphaFold 3. So we took amino acid sequences. We didn't even give structural data. Amino acid sequences of the toxins, fed it to AlphaFold 3 and asked it to predict the structures of these toxins. Of course, AlphaFold 3 is trained on also structural data. So there are toxins that are crystallized. So it has a good ability to predict the conformations there. And then we use these predicted structures to train now what is it called, RF diffusion and protein MPMN and NN, to then say make a binder to these toxins, and we could even tell it where to make the binders. So it was all done in silico, of course, then tested in vitro, and we selected the best neutralizers, and then also demonstrated that yes, against the toxins we wanted them to neutralize, they did have neutralizing capacity.
You don't need to say more, but the perfect example that in this case AI is working. For example, we have tried to adjust our SARS-CoV-2 antibody, which was in clinical trials, and we failed later with Omicron because we have some mutations where we know that we cannot bind, like CAR417N or CAR417T, and then we made, let's say, two ways: HI, human intelligence, and AI. And
in my lab, we use PI. Great PI.
But finally, human intelligence was better. And we made all the serine, we measured the affinity, and sometimes AI said the difference. So they said we will get a decrease of delta delta G, and AI said we're getting the opposite. So there's still a lot of things to do in this field. And your and Timothy's paper is a good example of what is possible. But it needs time that you will come for antibodies. Regarding production system, you're also going for E. coli. Does it make sense? CHO and E. coli, you also have to make all these LBS checks and.
So I think if you talk about human monoclonal antibodies full length, it will be sure.
So
if you go with the nanobodies, I think I would hardly say traditionally because not so much manufacture exists, but generally if you talk about therapeutic VHHs or therapeutic nanobodies, single domain antibodies, it's mostly done in PK yeast for a mix between relatively high yields, relatively simple manufacture. And I think it's AppLinks that sort of started doing that, and then since they started, then everyone kind of does the same.
There's also one antibody on the market, but this is also VHH tandem, to my knowledge.
Well, there's actually 4 now.
Blood coagulation disease.
Yeah, Caplacizumab, that was AppLinks. That was the first one, but there's actually 3 since then.
Okay, then
last year, 2 from China, or was it the year before? But
this one?
but I looked it up recently because I usually said there's only 1, and then suddenly there was 2, and now there's 4. I can't remember if they're all these divalent constructs. Maybe Caplacizumab is. I wouldn't necessarily put all my trust in E. coli, and I'm actually skeptical for the reasons you mentioned. They also tend to make inclusion bodies, and listened a lot to my dad, who's been working in manufacturing, who hates E. coli. But beggars can't be choosers, so when some quite brilliant Austrian scientists approached me and asked if they could test my nanobodies, or they wanted to do something else. And then I said, no, you should actually test my nanobodies, because if you can show that your system works with nanobodies, they will work with any nanobody. And that has relevance for you as a company if you look beyond snakebite. And they agreed on that. What they told me, they could reach really high yields and that they could get the E. coli to secrete proteins, which is difficult and not something you typically see. And I mean, I was skeptical, but the work I highlighted, led by Anne and Patrick, has shown, I didn't show the yields here, but without any specific optimization. So of course N-genes have optimized their general cell line, but we've just put it in our different antibodies, and we reach, now I forgot the lowest one, but between 12 and 17 grams per liter. So really high. Yeah, and more than half of it is secreted, if I remember correctly. In some cases, it's 3/3. And one could imagine that maybe improving a few things, maybe titers could go even higher, or secretion systems could be boosted even more. So, I mean, if you get to those levels, then you could argue that yields for a pharmaceutical product then matter less. You're right about the LPS. They show that it can be removed, and I mean, E. coli is used for pharmaceutical products.
For many other
The benefit is that it's grass, so you could maybe more quickly get it through regulatory, although you have to prove in your downstream that you get rid of LPS and so on. But and when I say beggars can't be choosers, I mean, I don't have $1 million or $5 million to set up a GMP manufacturing. So if someone wants to do it for free, I'm all in.
I'm all. Okay. You have also shown a very good example of affinity maturation. You use light chain shuffling, which is possible, and you have also shown, I remember, you have also shown that this is possible. I'm always a skeptic a little bit because of light chain shuffling normally, because the main affinity is coming from the heavy chain. Light chain in some cases is only stuffer, of course, and it's also good to make better light chain. What other possibilities do you have, and why do you say I do think light chain and not other possibilities?
So the reason we used light chain in the study I highlighted was also that John McCafferty and Anish and I, we speculated that maybe this is just enough, because typically they had seen from a lot of experience that you would get 10x more affinity, that maybe that's enough to just push it over. And it was relatively quick and easy compared to going in with the more heavy cavalry and doing either directed or semi-directed mutations in the CDRs, which would of course be, I think, the most obvious alternative to do that.
We are also happy normally with completely random mutations.
You could say this is maybe a perceived benefit. If you do light chain shuffling, you actually retain having fully human chains. Right. Whether that's actually a benefit or a perceived benefit, but at least it sounds nice that it's fully human and you haven't introduced.
Or you can set it as fully human.
You can. Yeah, that's what I mean. If
know what's coming.
That's what you could argue, right?
Okay, what do I else have? Models. This was in a spitting cobra. You have mainly used phage display, of course, because you're working together with John McCafferty. But what do you think about single B cell cloning, which is often used, especially for making anti-cancer antibodies, when you have these patients in these countries directly taking their blood, making single B cell cloning? Of course, you can also make an immune library of the single patient, but single B cell cloning is also in industry a procedure which they really like. Why not?
It could be done. I would say the complications with it is, of course, first of all, finding these people. Someone has to fly to Africa, find them, convince them, translate, get ethical approval, all that stuff. I would say whether it's more or less important, but at least equally important is, we know from the horses when you want to make an antivenom that you need to immunize horses repeatedly for a year and a half to get the antibody titers strong enough. So if you look at a patient who's just been bitten once, we've actually even seen
The immune answer is not.
Hmm?
You mean the immune answer is not as
it's not that much better than naive.
from viral diseases,
Yeah.
survivors, Ebola survivors, they have a very good immune answer. These groups, so it's
Yeah, no, I think that's true, but I also think many of the toxins, especially if you take neurotoxins, they're not very immunogenic. And I think it was my second paper in my PhD. We took blood samples both from someone who had challenged himself with venom, and someone, a Danish herpetologist, who accidentally was bitten by a snake. And of course, we based it on ELISA, which Brian and I just discussed. Is maybe not the perfect, but we could see that it was not crazy impressive, the amount of antibodies. Could it be that some of those antibodies turned out to be fantastic? Maybe. But I would say it was not like it looked like we would strike gold going for this. And then the complications involved are larger than actually just doing it with naive antibodies, I would say. But again, it's a viable strategy that has some benefits, that you don't need to deal with making libraries, which Sheeran tells me is a pain in the neck. But you have a different pain when you have to go out and go through clinical boards and ethical approvals, plus the logistics of getting blood.
Approval is also European problem. You will also show diagnostics on one of your last slides. And you showed a lateral flow strip assay. I'm not sure. What will you measure there? Will you use blood and then detect directly the toxin? Would be antibodies sensitive, or would the sensitivity of an antibody enough to measure these very
Yeah.
low concentrations in the
Yes.
bloodstream? So is it realistic or not?
We certainly,
not sure.
we certainly think it's realistic. And I mean, just to explain how it works, it's similar to the COVID test or the pregnancy test, where you have an antibody on a test strip. You have a sandwich antibody deposited where you add either blood or serum, could also add urine, or a wound swab in buffer that you then add. And then if there are toxins that are recognizing the antibody, they will bind and they will be transported over and then get stuck here. And why does it do that? That's how it works, and you'll see a clear line if there is a toxin. And I mean, we see it works. We developed on a few different prototypes, but one prototype we are testing in dogs and have already tested in two smaller, I couldn't call it clinical trials, but veterinary trials in dogs, where we do see that we can detect whether a dog is envenomed, also when we sort of blind test
material from patients.
Dog patients in this case.
What dog patient?
Dogs with 4 legs.
Dogs, okay.
The limits of detection we can reach, I think you can probably go a lot if you really engineer your antibodies, but with some optimization, we can reach around 1 nanogram, maybe 10 nanograms per milliliter. And in envenoming cases, in severe envenoming cases in dogs, you see 1,000 nanograms per milliliter.
Okay,
so
would be sensitive enough.
That's what we see. Of course, then it's a question of reducing false positives and false negatives and all sorts of stuff. And there's surprisingly much trouble on optimizing buffers, but it works.
Why are so much trouble?
I don't know.
I don't
No,
know.
I'm also disappointed with the antibodies.
Okay.
I mean, not those particular. It's general for all antibodies. Things behave different when they start absorbing to a surface. I mean, it works. And the thing is, it's about just optimizing salt concentration, sucrose, adding maybe a little bit of PEG to avoid unspecific binding, and yeah.
Okay. No, I'm also nearly finished. So it's, as I always say, it's very impressive. And thank you. And I think I'm coming back to Camilla.
But thank you, Michael.
Thank you so much, Professor Michael Hougaard. And at this point, I want to actually thank all opponents, our 2 opponents, and also our chairman of the assessment committee, because now the defense is concluded, and the 2 opponents and the chair of the assessment committee will withdraw and deliberate and make their recommendations. Of course, we are waiting excitedly, but while we wait, please join me in the lobby outside for a drink, and we'll see what the answer is. Thank you very much.
Thank you.